Hi everyone! You have successfully made it to week 5 of the Neuroscience and Relationship course. This is lecture 3, and I'm Julie Masasovic, the instructor. Today I'm going to focus on the limbic system. I'm spending a whole lecture on this particular system because it's of crucial importance to working in the field of psychology. Its role in emotions, drives, memories, and trauma make it essential to our work. My objectives for the lecture are as follows. As is my habit, I'll give you a little history of the limbic system and its discovery. Then I'll introduce the location of the limbic system and identify which brain structures are included. I will outline the functions of the limbic system, and then I'll go through these functions and explain in detail the roles of the various structures. I had originally planned to include trauma in this lecture because it's so closely related, but then decided that trauma in the brain needs its own lecture because of the bulk of information. So this lecture will simply foreshadow the one focused on trauma. It can feel a bit cumbersome. My hope is that by introducing the anatomy in this lecture, you'll be more familiar with and therefore not overwhelmed with all the new vocabulary when we start talking about trauma in the brain, addiction in the brain, etc. So let's get started with the history of the limbic system, which is really quite simple. Our old friend, Paul Broca, the French physician and anatomist, named this part of the brain le grand lobe limbique in 1878. Limbus, in Latin, means a distinctive border or edge. The limbic lobe is located on the medial edge of the cerebral cortex, and it surrounds the borders of the rostral brainstem, the corpus callosum, and the anterior commissure. Here's the picture. You can see the purple ring-like lobe, named by Broca, is located below the cingulate sulcus, sulci being the so-called depressions or fissures or valleys of the folds of the cortex of the brain. And the lobe surrounds the corpus callosum, the white part, and the upper brainstem, in gray. Then in 1937, an American physician named James PAPEZ was studying rabies, a disease that has the side effect of high levels of aggression. He observed that high levels of aggression were associated with damage to the hippocampus. As part of his experimentation, PAPEZ injected rabies virus into a cat's hippocampus and then monitored its progression through the brain. As a result of his observations, he made the conclusion that this particular circuit in the brain must be related to emotions. He developed an anatomical model of emotion, which he named the PAPEZ circuit. The circuit looks like this. Starting in the upper right corner of this diagram, fibers arising in the subiculum section of the hippocampal formation enter the fornix and travel forward to both the medial and lateral mammillary nuclei, aka the mammillary body. Then the circuit projects through what is called the mammillothalamic tract to, you guessed it, the anterior thalamic nucleus in the thalamus, onto the cingulate gyrus, then through the cingulum to the parahippocampal gyrus, and finally the entorhinal cortex, and full circle to the hippocampal formation. These structures you will learn to recognize by the end of this lecture, so I add them here just for exposure. Around the same time, Paul MacLean was also interested in the PAPEZ circuit. You may recognize MacLean as the man behind the triune brain theory. MacLean had also read Paul Broca's research, which indicated that the limbic lobe that surrounds the brainstem is a structure present in all mammals. He visited PAPEZ at Cornell University, after which he proposed a modified version of the PAPEZ circuit in 1952, emphasizing not only the hippocampus, but also the amygdala and septal area. He modified Broca's and PAPEZ's work and named the limbic system. The term limbic system includes a set of brain structures that are not limited to one lobe, but rather appear to cross into the frontal lobes and the temporal lobes. This picture is what it would look like if you cut the brain in half front to back and were looking at the inside of one half, otherwise known as the midline cut. The structures of the limbic system have evolved from a system developed mainly for the sense of smell in simpler animals. Today, The limbic system is thought to perform such diverse functions as regulating emotions, memory, appetite, and autonomic and endocrine control. At first, I didn't think our coloring book did a very good job with the limbic system, which will be part of our coloring assignment on week 7. It's Unit 5, Plate 26, if you want a preview. But then after doing my own extensive search for images of the limbic system, I realized how hard it is to find an accurate image. It's quite frustrating because different sources claim that the limbic system is made up of different parts. In fact, many resources inform me that there is no universal agreement as to which structures are official members of the limbic system. I believe the confusion lies partially in the fact that while we like to refer to main structures of the brain, the limbic system seems to be more a collection of parts of the brain. For example, certain nuclei in the thalamus are part of the limbic system. Also, certain parts of the cortex are part of the limbic system. So you will see varying opinions on this, but the following are generally identified as parts of the limbic system. The main parts are, starting at the image in the slide, clockwise from the right side, the hippocampus, the amygdala, the mammillary bodies, the hypothalamus, the olfactory cortex, parts of the thalamus, parts of the cortex, which include the cingulate gyrus, the subcolossal gyrus, and the paraterminal gyrus, the fornix, and the septal area. The septal area is often mistakenly called the septum, which is not shown in this image. In anatomy, a septum, which is Latin for something that encloses, is a wall dividing a cavity or structure into smaller ones. The septal area is rather a collection of neurons, which we will talk about later. The structures of the limbic system are evolutionarily ancient, and actually, the limbic system in many species takes up the majority of their forebrain. This image also isn't the greatest, but you get the point. The blue area is the limbic system viewed here from left to right in the human brain, the rabbit brain, the cat brain, and then the monkey brain. Its functions are also ancient, playing an integral role in survival. You could summarize the major functions of our limbic system into 4 basic categories. There are the functions which maintain homeostasis in the body, mostly associated with the hypothalamus. This includes controlling body temperature, hunger, important aspects of parenting and attachment behaviors, thirst, fatigue, sleep, and circadian rhythms. Then there are the olfactory functions, attributed to the appropriately named olfactory cortex, obviously involved in our sense of smell. Thirdly are the memory functions, associated with the hippocampal formation, aka the hippocampus. And then lastly you have the functions related to emotions and drives, famously associated with our amygdala. A mnemonic that might help you remember the 4 functions is HOME, or home, for homeostasis, olfactory, memory, and emotion/drives. This chart is, of course, an oversimplification, and there is a complex network of structures that are involved in all 4 of these functions. But these are the primary organs associated with these particular functions. So we'll start with the quick and easy, the olfactory function. I won't say a lot about the olfactory functions of the limbic system, but basically, axons from our olfactory sensory neurons converge in the olfactory bulb to connect the dendrites of other cells that send their axons to a number of brain areas, including, but not only, the olfactory cortex and the amygdala in our limbic system. Unlike other sensory systems, the olfactory neurons connect directly with the olfactory cortex without the delay of stopping over in the thalamus like all other sensory neurons do. The olfactory cortex includes the piriform cortex, that little orange jelly bean looking structure, and the periamygdaloid cortex, that little green jelly bean looking structure. The piriform cortex is probably the area most closely associated with identifying odor. The primary olfactory cortex projects to secondary olfactory areas. One of these is the anterior entorhinal cortex, the little blue structure which communicates directly with the hippocampus. This also plays a role in memory. This may explain why odors often have the ability to evoke strong memories. The connection of the olfactory cortex with the amygdala is involved in social functions, such as mating and the recognition of animals of the same species. We can think about pheromones and the way that dogs greet one another by smelling each other. The second function of the limbic system is the homeostatic functions, primarily associated with our hypothalamus. You may remember the hypothalamus from Lecture 2 as being part of the endocrine system. The hypothalamus, named for its location, which is below the thalamus, is a small group of cell nuclei, roughly the size of an almond, that perform a variety of functions. One of the most important of these roles being the link of the nervous system to the endocrine system via the pituitary gland, as discussed in the last lecture. The cell bodies that make up the hypothalamus have axons and terminals in the posterior pituitary gland. These terminals secrete the hormones oxytocin and vasopressin into the circulation. The hypothalamus is also responsible for release of hormones in the anterior pituitary by secreting certain inhibitory and releasing factors. These are a little confusing, so I'll show you a chart in a minute. It might be helpful to think of the hypothalamus in some ways as the upper management of the pituitary gland. It sends its messages to the pituitary gland, which then does its lower management work by sending messages to various other areas, releasing hormones. In this image, you can see how intimately related the two structures are. Thankfully, most of these names indicate exactly what they do, so there's not a whole lot of memorizing of complex names. Corticotropin-releasing hormone, vasopressin, and other peptides originating in the hypothalamus encourage the release of adrenocorticotropic hormone in the pituitary, which is involved in increasing the adrenocortical hormone, such as cortisol, which then increases blood glucose levels and inhibits inflammation and immune response. Thyrotropin-releasing hormone in the hypothalamus encourages release of thyroid-stimulating hormone in the pituitary, thereby increasing the secretion of thyroid hormones involved in metabolic rate, growth and development, and management of calcium in the body. Growth hormone-releasing hormone stimulates the release of, you guessed it, growth hormone in the pituitary, which stimulates growth via protein synthesis. Prolactin-releasing factor and thyrotropin-releasing hormone aids in the release of prolactin in the pituitary, stimulating milk production in the woman's breast. Luteinizing hormone-releasing hormone in the hypothalamus facilitates the release of luteinizing hormone in the pituitary, which plays a role in ovulation and progesterone production in females and the testosterone production in males. Luteinizing hormone-releasing hormone also facilitates the release of follicle-stimulating hormone in the pituitary, necessary for egg maturation and estrogen secretion in the female and sperm production in males. The hypothalamus also has some inhibitory factors, including at times inhibiting the growth hormone or prolactin release. So with regard to our endocrine functions, the hypothalamus is very active in many areas. Now the hypothalamus has other important functions outside of the endocrine system. In this picture, you can see that the hypothalamic nuclei can be divided into many different regions that have different functions. I will not outline all of those here, but I will say that the suprachiasmatic nucleus, shown in the picture on the right side as a small green egg-shaped structure, is considered the master clock of the brain, managing our circadian rhythms. Other non-endocrine related functions of the hypothalamus include important influences on both the sympathetic and parasympathetic divisions of the autonomic nervous system. Inputs to the hypothalamus that regulate autonomic functions include the amygdala and other regions of the limb. The limbic hypothalamic connection may be an important mechanism for emotional influences on autonomic pathways, explaining why your palms get sweaty and your stomach churns when you are anxious, as well as the emotional influences on the homeostatic pathways, such as the immune system. Perhaps this explains why depressed individuals may be more susceptible to infections. Positron emission tomography, or PET scans, and functional magnetic resonance imaging, fMRIs, have shown that the limbic system is one of the most active brain areas during the process of dreaming. The limbic system probably interweaves unconscious primal emotions with our conscious cognitive thoughts and perceptions, and thereby ties together emotions and memory during rapid eye movement sleep to form the content of dreams. GABA producing neurons in the hypothalamus contribute to non-REM sleep by inhibiting arousal systems, thereby promoting sleep. Therefore, certain lesions in the anterior hypothalamus have been shown to cause insomnia. Conversely, lesions in another area of the hypothalamus result in hypersomnia or an inability to stay awake. The lateral hypothalamic area, or LHA, contains orexinergic neurons that promote wakefulness. The orexinergic neurons inhibit the sleep-promoting mechanisms and the REM sleep-promoting neurons. Orexin, also called hypocretin, is a neurotransmitter that regulates arousal, wakefulness, and appetite. The most common form of narcolepsy, in which the sufferer briefly loses muscle tone, is caused by a lack of orexin in the brain due to destruction of the cells that produce it. The brain contains very few cells that produce orexin. In a human brain, about 10,000 to 20,000 neurons in the hypothalamus have this important job. However, the axons from these neurons extend throughout the entire brain and spinal cord, where there are also receptors for orexin. The hypothalamus is also important in appetite. Certain lesions in the hypothalamus have been related to weight loss, while other types of lesions result in obesity. Thirst also appears to be a result of activation in the hypothalamus, as is certain mechanisms involved in temperature regulation. The hypothalamus seems to be responsible for detecting elevated body temperature and then activating our cooling mechanisms, such as sweating and other areas that are used to conserve heat. Certain lesions have been shown to cause hypothermia. The hypothalamus probably participates in the circuitry involved with sexual desire, and in particular has been found to be a key structure in the central control of male sexual behavior, such as playing a role in erection and ejaculation. The hypothalamus may also play some role in complex motivational states. Now on to the mammillary bodies. The mammillary bodies are considered by neuroanatomists to be a part of the hypothalamus. They act as a relay for impulses coming from the amygdalae and hippocampi via the mammillothalamic tract to the thalamus as part of the PAPEZ circuit. Using MRIs, scientists have also discovered that sleep apnea patients' mammillary bodies were nearly 20% smaller, particularly on the left side. One of the investigators hypothesized that repeated drops in oxygen lead to the brain injury. Another disorder, known as Korsakoff psychosis, is caused by damage to mammillary bodies, the dorsomedial nucleus of the thalamus, and the hypothalamus. It is a syndrome associated with chronic, prominent impairment of recent and remote memory. Recent memory is characteristically more disturbed than remote memory. Immediate recall is usually preserved. Confabulation may be marked, but is not always present. So just to check in, I know this information can be daunting. Pause and take a break if necessary. Do some stretching and a little deep breathing, and then keep on going when you're ready. Moving on to the memory function of the limbic system. There are two main regions of the human brain that have been found to be critical to memory formation. One of these regions is in the limbic system. The key structures are called the hippocampal formation and the parahippocampal gyrus. Here's a somewhat sketchy picture of the hippocampus. I apologize for the picture quality. I actually could not find a clearer picture of a close-up of the hippocampal anatomy. But if you open your coloring book to Unit 5, Plate 27, you can get a look at their portrayal of the hippocampus. I wanted you to see the seahorse-like shape after which it is named, from the Greek hippocampus, meaning seahorse. That lower gray portion is the parahippocampal gyrus. and the upper curlicue portion, starting with the aqua color and on up, is the hippocampal formation. There are three components of the hippocampal formation: the dendate gyrus, the hippocampus, and the subiculum. Generally, when someone is talking about the hippocampus, they are referring to all three of these structures. The hippocampal gyrus is also made up of several different structures, but I think it's unnecessary to name them all here. It gets too technical. You may remember from your Psych 101 class the classic study of patient HM. In 1953, a 27-year-old man with the initials HM underwent an operation in an attempt to control epileptic seizures. The bilateral medial temporal lobes, including his hippocampal formation and the parahippocampal gyri, were cut. After the surgery, his seizures improved, but he had severe memory problems with no other significant deficits. He was unable to learn new facts or recall new experiences. So, when given a list of 3 or 4 words to remember, he was able to correctly say them back immediately, but within 5 minutes had lost them completely and couldn't remember that he was given the list to begin with. However, his memory of remote memories of his childhood and up to several years before the surgery were intact. Personality and intelligence remained normal, and he was able to learn certain tasks that did not require conscious recall. For example, his performance on a mirror drawing task improved on successive days, similar to normal controls, even though he could not remember doing the task the previous day. This landmark study of patient HM led to many experiments about the medial temporal lobe's role in memory. HM himself continued to participate in experiments for decades until shortly before his death in 2008. Patients today still get unilateral medial temporal lobe resection, but never bilateral, to treat specific forms of epilepsy. One of the important things learned from studies associated with HM was the difference between declarative memory and non-declarative memory. A little review. Declarative memory, also known as explicit memory, refers to those memories that are a conscious recollection of facts or experiences. For example, remembering your grocery list or what you did yesterday. Non-declarative, aka implicit memory, is the non-conscious memory of skills, habits, and other acquired behaviors such as walking, or remembering how to drive a manual transmission car, even after non-driving one for years. So HM suffered a loss of declarative memory, while his non-declarative memory was intact. The term amnesia generally refers to the loss of declarative memory loss. Another area of research as a result of HM's experiments allowed us to distinguish between different types of memory, such as working memory, which is recall within 1 to 2 minutes of learning. HM's working memory seemed to be intact. Recent memory is recall 4 to 5 minutes later. HM did not have recent memory after his surgery. And remote memory includes things such as previous addresses or schools or well-known current events. These types of memory are tested during a mental status exam when, for example, a patient first comes in for an assessment. Working memory can be tested by asking the client to repeat back lists of digits or words forward or backwards. Recent memory is tested by asking the client to remember 3 words and then asking a few other questions before asking them to recall those 3 words. And remote memory can be assessed by asking past addresses or schools attended or some other personal verifiable information. And a third thing we learned or studied as a result of HM was anterograde amnesia and retrograde amnesia. HM suffered from anterograde amnesia, which is the inability to form new memories. But he also suffered from retrograde amnesia in that he was unable to recall memories from a few years before his surgery up to the point of surgery. This combination of retrograde and anterograde amnesia for declarative memories is typical of lesions in the medial temporal lobe or medial diencephalic memory systems, although it can also be caused by concussion or other disorders. As a side note, the hippocampus has been implicated in the genesis of ADHD because children and adolescents with combined type ADHD have been found to have enlarged hippocampi. A 2006 study of the MRIs involving 114 youth ages 6 to 18 showed that those diagnosed with ADHD showed larger hippocampi compared to a control group. Although conventional measures did not detect significant differences in amygdala volume, surface analyses indicated the presence of reduced size bilaterally over a portion of the amygdala. Correlations with prefrontal measures suggested abnormal connectivity between the amygdala and the prefrontal cortex in the ADHD group. The authors concluded, quote, "The enlarged hippocampus in children and adolescents with ADHD may represent a compensatory response to the presence of disturbances in the perception of time, temporal processing, and stimulus seeking associated with ADHD. Disrupted connections between the amygdala and orbitofrontal cortex may contribute to behavioral disinhibition. Our findings suggest involvement of the limbic system in the pathophysiology of ADHD." Now, moving on to the best known function of the limbic system, and arguably the one most applicable to our work in counseling, the emotions and drives function. Now, before we get to the amygdala, let me briefly cover the septal area, or septal nuclei, seen in the bottom right of this image. The septal nuclei send and receive connections from the olfactory bulb, the hippocampus, the amygdala, the hypothalamus, the midbrain, the habenula, associated with our pineal gland, the cingulate gyrus, and the thalamus. The medial septal nucleus contains cholinergic neurons, which are neurons that transmit acetylcholine, that project to the hippocampal formation and may play a role in memory function. The septal area is also considered a pleasure zone in animals, playing a role in reward and reinforcement along with the nucleus accumbens. In the 1950s, researchers Olds and Milner showed that rats with electrodes implanted in this area will self-stimulate repeatedly by pressing a bar to receive electrical current that will stimulate these neurons. The role the limbic system plays with regard to emotion and drives primarily lives in the amygdala. Amygdala means almond in Greek. The amygdala, which is officially named the amygdaloid nuclear complex, is a group of cell nuclei which can be separated into three main groups. The basolateral nucleus is largest and predominantly involves indirect and indirect connections of the amygdala to different regions of the cortex, as well as to the basal forebrain and medial thalamus. The corticomedial nucleus makes connections involved in olfaction and also interacts with the hypothalamus related to appetite. And the central nucleus is the smallest and has connections with the hypothalamus and brainstem that are important for autonomic control. A researcher named KLUVER in the 1930s was researching the area of the brain affected by the drug methylene when he observed the functions of the amygdala. He had neurosurgeon BUCY bilaterally remove the temporal lobes of monkeys. Among the effects observed were visual agnosia, an increased tendency to explore items by the mouth, the dampening of emotional expression, altered sexual behavior, and differences in diet. KLUVER later discovered similar observations had actually been published in 1881, which substantiated his own observations. The monkeys in the study seemed to have an inability to recognize the emotional importance of events. They did not display fear for items that would ordinarily frighten members of their species. They displayed an appetite for unusual foods such as rocks or live rats, and they sought intercourse with unusual partners, including members of other species. They became extremely interested in exploring items in their environment and became placid when approached. Similar symptoms became known as KLUVER-BUCY syndrome and were first documented among certain humans who had experienced temporal lobectomy in 1955. KLUVER-BUCY syndrome is now identified as being a result of the bilateral lesions of the amygdala. Its symptoms include docility, characterized by diminished fear responses or reacting with unusually low aggression. Dietary changes and/or hyperphagia, characterized by eating inappropriate objects, pica, and/or overeating. Hyperorality, defined as an oral tendency or compulsion to examine objects by mouth. Hypersexuality, characterized by a heightened sex drive or a tendency to seek sexual stimulation from unusual or inappropriate objects. And visual agnosia, characterized by an inability to recognize familiar objects or people. Other studies, both in animals and humans, have allowed us to make various conclusions about the amygdala's role. I'll share some of these conclusions here, but keep in mind that while the amygdala has been identified as playing a central role, it is only one part of a complex brain system that is responsible for emotions and drive. So as I go through all of this information, it is not exclusive to the amygdala. The role of the amygdala makes sense when you break down the purpose of emotions. Emotion seems to have the purpose of helping us to respond to events in the world with a particular focus on critical information that has import to us. Emotions motivate us to shape our behavior, to gain what we desire and avoid what we fear. So in other words, the amygdala seems to be important for attaching emotional significance to stimuli perceived by our association cortex, which is that part of the brain responsible for processing input from our senses and translating that into behaviors. This includes attaching emotional significance to memory. So you could see how integral the amygdala would be for traumatic memories, and therefore for working with traumatized. Now you probably already know that the amygdala is also thought to be related to our emotional responses of fear, anxiety, panic, aggression, and rage. Animal studies have shown that stimulating the amygdala appears to increase both sexual and aggressive behavior. Likewise, studies using brain lesions have shown that harm to the amygdala may produce the opposite effect. The amygdala also plays a role in autonomic behaviors with the support of the hypothalamus and the brainstem. It helps mediate changes in heart rate, peristalsis, gastric secretion, goosebumps, sweating, and other changes often associated with strong emotions. It helps activate our sympathetic nervous system and mobilize the body's fight-or-flight response. The amygdala also plays a role in olfaction and pheromone processing. It receives input from the olfactory bulb and the olfactory cortex. It is also involved in neuroendocrinological changes associated with emotional states. The amygdala is thought to be associated with odors and sense of panic related to the auras that occur prior to or independently from seizures. Studies have shown subtle bilateral decreases in the volume of the amygdala, hippocampal formation, and parahippocampal gyrus in schizophrenic patients. And there is some evidence that the amygdala is involved in anxiety disorders. Some studies have shown children with anxiety disorders tend to have a smaller left amygdala. In the majority of cases, there was an association between an increase in the size of the left amygdala and the use of SSRI medications or psychotherapy. The left amygdala has also been linked to social anxiety, obsessive and compulsive disorders, and post-traumatic stress, as well as more broadly to separation and general anxiety. Some have suggested that anxiety disorders may be a result of a failure of the anterior cingulate and hippocampus to modulate the activity of the amygdala. There is also some speculation about the amygdala playing a role in autism. Research performed by Bacavallere at the University of Texas Health Science Center looked at infant monkeys who had had their amygdalas lesioned. The monkeys exhibited abnormal social skills, such that they, quote, will not initiate in social approach as young babies normally do to play together, and they also seem to have ritualistic behavior like rocking. These behaviors remained when they became adults, end quote. Bacavallere believed that the damaged amygdala robs the young animals of their ability to interpret the social world around them. She said, quote, I have the feeling that these animals have a hard time interpreting facial expressions or any type of gesture the monkeys can have. Thus, they react as trying to avoid interactions. Another interpretation of the monkey's behavior might be that they are just frightened, which could explain the social deficit. Autism and Asperger's syndrome involve a disproportionate impairment in specific aspects of social cognition. Limbic structures involved include the cingulate gyrus and amygdala, which mediate cognitive and affective processing. The basolateral circuit, integral for social cognition, is disrupted in autism spectrum disorders. Borderline patients have also been found to have smaller amygdala. Amygdala plays a role in food choice and emotional modulation of food intake. The lateral nucleus of the hypothalamus is the center for control of feeding, whereas the ventromedial nucleus functions as our center of satiation. The reward circuitry underlying addictive behavior includes the amygdala and nucleus accumbens. The amygdala plays a central role in cue-induced relapse. Relapse associated with cues, stress, and a single dose of a drug of abuse results in release of excitatory neurotransmitters in the hippocampus and amygdala. So, the amygdala is thought to play a role in the motivation to take drugs of abuse and the compulsive nature of drug taking. Finally, the amygdala and the cingulate gyrus are involved in social cognition. In addition to what was said before regarding autistic behaviors, social cognition refers to thought processes involved in understanding and dealing with other people. Social cognition involves space perception, emotional processing, theory of mind, self-reference, and working memory. Together, the functioning of these regions would support the complex behaviors necessary for social interactions. Now, I promise this is the last structure we're going to look at. So last but not least, let me introduce you to our thalamus. The thalamus is made up of sections of masses of cells. We'll color the thalamus on week 6, but if you want a preview, turn to the coloring book, Unit 5, page 16 through 19. The thalamus is like Grand Central Station for sensory input. Almost all incoming information passes through the thalamus before going to the neocortex. The exception, of course, as discussed earlier, is olfactory sensory information. Thalamic nuclei that are generally agreed to be involved in memory, emotion, and arousal are the medial dorsal nucleus, the anterior nucleus, and the lateral dorsal nucleus. These nuclei have intimate connections with cortical and subcortical areas that are considered part of the limbic system of the brain and sometimes referred to as the limbic thalamus. They have also been termed the visceral thalamus because they are vital for maintenance of circadian rhythms and for the generation of normal sleep patterns. So the medial dorsal nucleus has many connections with various parts of the brain. These connections and the effects of injury to this region indicate a role in memory, perhaps specifically in retrieval of episodic memory, mood, motivation, and the sleep-waking cycle. It has been proposed that the connections of the medial dorsal nucleus segregate into 3 major functional circuits. One portion, with connections to and from the anterior cingulate cortex, is involved in motivation. Another portion has reciprocal connections with the orbitofrontal cortex and is involved in inhibition of inappropriate behavior. And the third portion has reciprocal connections with the dorsolateral prefrontal cortex and is involved in the mediation of executive functions. The anterior nucleus has major reciprocal connections with anterior and posterior cingulate cortices and parts of the hippocampal complex. Lesser connections with orbital frontal, dorsolateral prefrontal, and parietal cortices have also been reported. The anterior nucleus also receives major input from the mammillary complex. It may also receive a projection from the septal nuclei. These connections and the effects of injury to this region indicate a role in memory, modulation of the sleep-wake cycle, and directed attention. And lastly, the lateral dorsal nucleus of the thalamus has major reciprocal connections with posterior cingulate and parietal cortices, as well as the subiculum, presubiculum, and entorhinal cortex portions of the hippocampal complex. It may also receive a projection from the septal nuclei. A role in integration of motivation and/or attention with sensory processes has been suggested by research. A common source of injury to the thalamus is stroke, otherwise known as cerebrovascular accident or vascular insult. Injury to the left is associated with deficits in language, verbal intellect, and verbal memory. Right-sided injury is associated with deficits in visual, spatial, and nonverbal intellect, and visual memory, as well as delirium. Medial thalamus appears to be particularly important for temporal aspects of memory, and bilateral injury is associated with severe memory impairment called thalamic amnesia, as well as dementia. Some researchers attribute the memory deficit to destruction of the tracts connecting the limbic structures to the thalamic structures. Injury to the anterior and medial thalamus can also result in disturbances of autonomic functions, mood, and the sleep-waking cycle. Symptoms usually lessen with time, but commonly significant impairment remains. The limbic areas of thalamus are also vulnerable to non-vascular insults. Korsakoff syndrome results from thiamine deficiency, often as a result of alcoholism. It is characterized by both anterograde and retrograde amnesia, particularly for recent events and confusion. These symptoms have been attributed variously to involvement of the mammillary bodies and the limbic thalamus. Fatal family insomnia is a rare genetic disease characterized by severe neuronal degeneration in the limbic thalamus. Clinically, there is progressive impairment of the sleep-waking cycle, progressing to total loss of sleep, or insomnia, as well as autonomic dysfunction and gradual loss of hormonal circadian rhythm. Lack of sleep leads to hallucinations, delirium, and confused states like that of dementia. There is no known cure. The clinical features may be a direct result of neuronal injury in the limbic thalamus, or may result from disconnection of the hypothalamus, limbic, and prefrontal cortices. Alright, so I apologize for the length. Each week I think I'm not going to possibly have enough information to cover a half-hour lecture, and then I totally bombard you with lots of information. So I hope you were able to take some of that in. I really appreciate your attention, and we'll talk about it more in the discussion forum. Thanks so much. And last but not least, here are the references used for this lecture.